Dear students, welcome to EPG Pathsala. I am Dr. Y.S. Rajput, former Head of Division, National Dairy Research Institute, Karnal. Today, I am going to explain you one of the electrophoretic technique titled Agarose Gel Electrophoresis under the paper Biochemical Techniques. I have to remind you that we have modules on electrophoresis basic concept and acrylamide gel electrophoresis. I advise you to learn these modules prior to this module. As discussed in these modules, and I again repeat that electrophoresis is a separation technique where charged particles are separated in an electric field. The migration rate of charged particles depend on voltage, distance between electrodes, charge on particles, and frictional force. Frictional force retards the movement of charged particles and is generated from poles of support and from buffer. In SDS space electrophoresis, charge to mass ratio on proteins is constant and difference in frictional force exerted on different molecules leads to their different velocity in an electric field. We have discussed this technique in depth, and also it should be clear that SDS space is usually used for separation of protein and is performed in vertical gel electrophoresis unit. For electrophoresis, different supports are available. Nowadays, two supports are commonly used. These are acrylamide gel and agarose gel. This has resulted in coining of techniques wherein name of the gel itself incorporated. This is how the name of agarose gel electrophoresis is derived. I may explain that these are two parallel electrophoretic techniques and highly specialized techniques. Today I will be presenting in my presentation the agarose gel electrophoresis, and the focus will be on to make you understand principle of agarose gel electrophoresis. You will be also given a comparative aspect of sodium hydroxide sulfate polyacrylamide gel electrophoresis vis-a-vis agarose gel electrophoresis. The presentation will also cover how to perform agarose gel electrophoresis. I will also be explaining the applications of agarose gel electrophoresis. Agarose is one of the two principal components of agar, which was discovered in 1658 by Mino Terojamon from Japan. Agar is mixture of polysaccharides isolated from seaweeds such as Gelidium and Gassileria. Agarose is a linear polysaccharide which contains agarose as repeating unit. Agarose is a disaccharide and consists of D-galactose and 3,6-N-hydro-L-galactose. So it should be clear that agarose is a fractionated from agar. In continuation to my introduction to the agarose, I have to state that molecular weight of agarose is variable and is in range of 80 kilodalton to 140 kilodalton. The most chance that the molecular weight of the agarose, it will be around 120 kilodalton. Agarose solubility in water is low. However, when aqueous suspension of agarose is boiled, it results in clear solution. When boiled and clear solution of agarose is slowly allowed to cool, it forms gel. Gel formation is ascribed to formation of intra and intermolecular hydrogen bonding. Intra-hydrogen bonds are within the long agarose chain, while intra-hydrogen bonds are formed between chains. Pore size of gel is dependent on agarose concentration. Gel formed from higher agarose concentration will be of low pore size and vice versa. Although agarose is supposed to be free from charge, sulfate groups are present at alternate sugars to a variable degree. Support matrix for electrophoresis should be free from charge. Sulfate group in agarose interferes in electrophoresis. This is explained in terms of electroendosmosis phenomena. Cations from buffer ions or salts can bind to sulfate and make electrical double layer. During electrophoresis, when voltage is applied, cations from electrolyte double layer moves towards cathode and in this process carry electrolyte solution towards cathode. This is referred as electroosmotic flow in the direction of cathode. The quality of agarose is determined by level of sulfate. Agarose with minimum level of sulfate is used in agarose gel electrophoresis for better separation. Agarose gel is used for separation of nucleic acids and proteins in electrophoresis. Only large size proteins can be separated in agarose gel electrophoresis. Immunoelectrophoresis is good example for separation of antigen on agarose gel prior to diffusion of antigen and antibody through agarose gel. In isoelectrofocusing also, agarose gel is used. It is true that electrophotic separation of nucleic acid is mostly achieved on agarose gel. It is pertinent to discuss the pore size of acrylamide gel and agarose gel. Although pore size of acrylamide gel and agarose gel is dependent on their concentration, in general, pore size of agarose gel is higher than acrylamide gel. Also, in general, size of nucleic acid is much larger than size of protein, both in acrylamide gel electrophoresis and agarose gel electrophoresis. Protein or nucleic acid is required to move through pores of support, and pores must create differential frictional force in movement of charged particles. This is the reason why proteins are mostly separated on acrylamide gel while nucleic acids on agarose gel. Of course, Small size nucleotides can be separated in acrylamide gel electrophoresis. Comparatively, elasticity of agarose gel is poor in comparison to acrylamide gel. It is difficult to remove agarose gel from a small tube or glass plates or glass cassettes after vertical gel electrophoresis. Thus, agarose gel electrophoresis is not performed on vertical electrophoresis unit and is carried out in horizontal electrophoresis unit. In both agarose gel electrophoresis and HDS space, separation is based on size of nucleic acids and protein respectively. In both the case, charge to mass ratio of respective molecules is constant. In HDS space, this is achieved by binding of HDS to protein, which acquires net negative charge. The binding of HDS to protein is almost constant for wide range of proteins, and for every two residues of amino acids in protein, one molecule of HDS binds. HDS binds to hydrophobic pockets in protein, and in doing so, sulfate groups are exposed out. Nucleic acids do not require any treatment like the one given to protein for making their charge to mass ratio constant. Nucleic acids have negative charge, and amount of net negative charge is proportional to size of nucleic acids. So, charge to mass ratio of nucleic acid is constant. Large size nucleic acids move slowly, whereas small size nucleic acids will move faster in agarose gel electrophoresis. Size of nucleic acid and size of the pore in acrylamide gel determine magnitude of frictional force exerted on nucleic acids in their movement. This situation is similar to HDS space. Pore size of agarose gel is dependent on concentration of agarose. Size of nucleic acids should be such that these are able to enter pores of gel, and all components in mixture should experience variable frictional force to allow their separation. Different types of agarose are available in market. These include agarose with characteristics of low electroendoosmosis, referred as EEO, medium EEO, and high EEO. High gelling temperature, low gelling temperature, and high gel strength. High gelling temperature gives higher thermal stability with elasticity and flexibility to the gels. Low gelling temperature agarose is preferred for chlorine experiments for extraction of small size nucleic acids. Shape of the molecule also affects the migration of molecules in an electric field. Molecules having globular shape will move faster than linear or fibrous shape. When plasmids subjected to resolution in electrophoresis, supercoiled plasmid will move faster. followed by coiled plasmid, and then by linear plasmid. Optimum concentration of agarose is required for separation of nucleic acids. The concentration of agarose depends on the size of DNA fragments which are to be separated. Molecular weight of nucleic acids is in general much larger than proteins. Size of protein is represented in terms of its molecular weight and denoted by Dalton or kilodalton. However, for nucleic acids, the size is represented in terms of base pairs or by BP, or nucleotides represented by NT. Size of double-stranded nucleic acid is denoted by base pairs, that is by BP, whereas size of singular-stranded nucleic acid is expressed in terms of nucleotide. 1 kilo base pair or 1 kb is equivalent to 6,20,000 Daltons. Commonly used plasmid, that is PBr 322, is of 24 lakhs Dalton, and the size of it is referred as 4.32 kilobase. Pore size or the sieving effect of agarose depends upon concentration of agarose. Generally, molecular size of above 50 base pair can be separated easily. Mobility of nucleic acid slows down in gels prepared with higher concentration of agarose. Sample resolution time is inversely proportional to concentration of agarose. Voltage or potential gradient also affects the migration rate. Migration of ion in an electric field depends on potential gradient, which is equal to applied voltage divided by the distance between two electrodes. It also depends on charge on particle, also on frictional force exerted by medium on particle. Higher voltage will increase current, which will result in more heat generation. Generated heat is to be efficiently dissipated, otherwise it will lead to poor resolution and broadening of bands. Dear students, we have covered fundamental differences in sodium dodecyl sulfate polyacrylamide gel electrophoresis and agarose gel electrophoresis. Both the techniques rely on separation of molecules based on their size. Different size of molecules create differential frictional force in movement, and this results in separation. Further, I am repeating that in general, size of nucleic acid is larger than that of protein. Also, pore size of agarose gel in general is larger than the pore size of acrylamide. Matching of size of macromolecules with pore size of the gel matrix is essential for separation of macromolecules in an electric field. This is the reason why, in general, proteins are separated on acrylamide gel, while nucleic acids are separated on agarose gel. Now I will be explaining how the agarose gel electrophoresis is performed and what are the applications of agarose gel electrophoresis. Now I will be explaining how the technique of agarose gel electrophoresis is performed. For this, we require certain equipments which include power supply, which can give output in two different modes, either in constant voltage or constant current. We also require horizontal electrophoretic unit, which is available in variable size for variable number of samples. In this electrophoretic unit, from different company or from the same company, different models will have the D value, that is distance between two electrodes, different. We also require casting unit. Also we will require transilluminator, which can illuminate the gel under ultraviolet light. Besides the equipment and accessories, we require a number of consumables for performing Agarose Gel Electrophoresis. These includes chemicals, buffers, and dye solutions. Agarose of desired specification is required for gel preparation. We require DNA sample. We require nucleic acid markers or DNA ladders, tris base, glacic acid, boric acid, EDTA, disodium salt of EDTA, ethidium bromide, xylene cyanol dye, bromophenol dye, and sodium dodosol sulfate, glycerol. Low ionic strength as well as relatively high ionic strength buffers is used in Agarose Gel Electrophoresis. Low ionic strength buffer is popularly referred as tris acetic acid EDTA or TAE buffer, which essentially contains 4 millimolar tris, 1 millimolar EDTA, and the pH of buffer is adjusted to 8.0 with acetic acid. This buffer is used for separation of high molecular weight double-stranded DNA. The purpose of having EDTA in a buffer is that it sequesters divalent ions, which are essential for nuclease action. This buffer is also preferred for DNA recovery. The other buffer, which is commonly used, is referred as tris-borate EDTA of pH 8.3, and in short it is called TBE buffer. It is comprised of 89 millimolar tris, 89 millimolar boric acid, and 2 millimolar EDTA sodium salt. Ionic strength of TBE buffer is higher than TAE buffer. In low ionic strength buffer, nucleic acids carry more current and migrate faster. Borate is inhibitor for many enzymatic reactions, and thus TBE buffer is to be used with careful decision that borate can inhibit many of the enzymatic reactions. In that situation, the use of borate buffer is to be avoided. Both buffers inhibit protonation of DNA and protect DNA molecules from enzymatic degradation. In this, we will be explaining the purpose of having the loading buffer. Loading buffer is used to dilute the sample before loading. The purpose of this buffer essentially is to increase density of the sample and its visibility on loading. Movement of dyes present in loading buffer provides a status of electrophoretic run. Loading dyes are negatively charged in buffer, and hence these will migrate in the direction of DNA migration. Stock solution of 6x sample buffer, that is the loading buffer, contains 30% glycerol in distilled water, 0.25% bromophenol blue, and 0.25% xylene cyanol dye, and is prepared in simple buffer, which should be of low ionic strength than electrode buffer. High ionic strength of loading buffer causes bands to be fuzzy and results in migration of DNA at unpredictable rates. Alkaline loading buffer is used for alkaline gel electrophoresis. Molecular weight markers, or referred as DNA ladder, is also dissolved in loading buffer. These essentially contain mixture of DNA molecules whose molecular size is known. Ladders are available in size difference of 100 base pair or smaller, or 1000 base pair. Size of bacteriophage lambda DNA is 49 kilobase, which on action of restriction enzyme generates fragments of 21.80 kb, 7.52 kb, 5.93 kb, 5.54 kb, 4.80 kb, and 3.41 kb. These fragments can also be used as molecular markers. These are loaded to one of the well in Agarose to allow correct determination of size of DNA in sample. For performing the Agarose Gel Electrophoresis, agarose gel is to be prepared and it has to be casted in casted urine. For this purpose, agarose is dispersed in 1x TAE or TB buffer, as explained earlier, without clumps in clean glass conical flask to obtain required concentration of gel. The contents are heated in microwave oven or on hot plate. When solution becomes clear and transparent, it is allowed to cool to approximately 55 degrees centigrade. At this stage, ethidium bromide is added to obtain 0.5 microgram per ml concentration in gel, and the contents are swelled without forming any air bubbles in melted agarose. Immediately, transparent agarose gel, while it is hot, is gently poured in gel casting unit, avoiding trapping of air bubbles. Depth of the gel can be kept to 3 mm. Ethidium bromide is a carcinogenic compound and thus it requires careful handling, which includes use of lab coat and gloves. Comb is then placed gently in gel at appropriate location. When gel is solidified, gel is ready for electrophoresis. When gel is ready, it is solidified, then electrophoretic run is to be initiated. So we say that when gel and samples are ready to use, electrophoresis unit tank is filled with 1x TAE or TB buffer to a height to allow merging of gel in buffer. Then gel is placed in electrophoretic unit, comb is removed without any damage to the well, sample is loaded in wells created by the comb in gel by using dispenser. Molecular markers or the DNA ladders are usually loaded in first or the last well. Electrophoretic chamber is then covered with lid. Electrophoresis is done at voltage of 5 volts per centimeter. That is the distance between the two electrodes. It is essential to connect leads to power supply correctly. Movement of DNA and markers will be towards anode. Edge effects will be observed when high voltage is applied. The electrophoretic run is continued till loading die migrates to appropriate distance. After the electrophoretic run, the time will be to see the bands on separation. Nucleic acids are visualized by using ethidium bromide. Ethidium bromide intercalates the major groups present in the DNA. Its density enhances wherever the DNA in gel is there. When gel is exposed to UV light, ethidium bromide emits fluorescence and DNA appears as orange-red. DNA can also be stained with ethidium bromide after electrophoretic run, and this requires placing of gel in ethidium bromide solution for 5 to 10 minutes. 10 nanogram of DNA can be detected. Other alternative dyes to visualize the DNA are also available. This includes cyber green, which is used for quantification of nucleic acid. This dye can also label DNA within the cells. Other dyes, such as gel lead, is closely related to ethidium bromide in its structure and binding, but it is less toxic and more sensitive. Once the dye is stained with ethidium bromide, DNA fragment can be visualized under ultraviolet illumination. Under ultraviolet light, aromatic ring of ethidium bromide molecules activate and release energy in the form of light. Generally, ethidium bromide absorbs in the range of 302 or 312 nanometer, known as UV-B rays. Longer time exposure of DNA to UV light can damage DNA and thymine dimers can fall. If DNA is to be recovered, it should be minimally exposed to UV light. Visualization at 365 nanometer, called UVA, causes less damage to DNA, but visualization is poor. It is essential to protect one's eye by wearing goggles when UV light is used. While viewing the gel exposed to UV light, UV shields must be placed over the gel for protecting eyes. Agarose Gel Electrophoresis is a separation technique, and this technique is having wider use in molecular biology research. I will be explaining some of the applications where this Agarose Gel Electrophoresis can be performed. As I said a number of times, Agarose Gel Electrophoresis is a technique for separation of nucleic acids. This technique is used for evaluation of PCR, that is the polymerase chain reaction. Polymerase chain reactions has wider applications. We name any field, we find the application of PCR, whether it is for resolving the issues of crime, whether it is for resolving the issues of paternity, whether it is required for cloning an expression of a particular gene for making a desired product, whether it is required for targeting a gene, any applications we name for the PCR reaction. This Agarose Gel Electrophoresis is always used in continuation of PCR reaction. So we cannot bifurcate the application of Agarose Gel Electrophoresis from the polymerase chain reaction. In nutshell, what we will say that the applications, all applications known for the PCR reaction are in fact evaluated by the Agarose Gel Electrophoresis. Besides this particular aspect, Agarose Gel Electrophoresis is also used for separation of the nucleic acids in general, also to any restriction fragments used for the cloning purpose in particular. And also we see that this is a technique by which it is possible to find out the size after action of restriction enzymes on the nucleic acids. We can also isolate the fragments on gel after electrophoresis for cloning purpose. We name any applications for molecular biology, we find that this particular technique is very well linked with the molecular biology work. With this, we will say this is very highly specialized technique, but of course, the technique can be performed by a technical person only. This is, we say, could be a limitation. This is not a simple technique which any person with a moderate knowledge of the science can do it. It has to be performed by a specialized technical hands for its success. Dear students, let us summarize the topic of discussion. Agarose Gel Electrophoresis is a specialized electrophoretic technique. You cannot think that lab working on polymerase chain reaction is not routinely using this technique. Agarose Gel Electrophoresis is widely used in labs focusing their research on nucleic acids. For detection of bands in gel, ethidium bromide is required. This chemical is hazardous and should be handled carefully. Disposal of waste containing hazardous material must be made by taking care guidelines available on the subject. Visualization also requires exposing the gel to ultraviolet radiation, and thus user must protect himself or herself, especially eyes and skin. Dear students, while performing Agarose Gel Electrophoresis, you must be careful in selecting desired purity of agarose. In none of the situation you allow generated heat damage the experiment. You should also be careful in not contaminating buffers with nucleic acid hydrolysis, otherwise these will degrade DNA. Now you must be very clear that why people are using Agarose Gel Electrophoresis in studying the nucleic acids. Further, I want to make you very clear in the form of summary that size of nucleic acids is larger than the size of proteins. The pore size of agarose gel is larger than pore size of acrylamide gel. Also, I want to make very clear that if we change the concentration of acrylamide or we change the concentration of the agarose gel, then pore size changes. Higher the concentration of the gel, lower is the pore size. If lower is the concentration of the gel, higher is the pore size. Because of this pore size, even it is possible to use the Agarose Gel Electrophoresis for separation of large size proteins. Similarly, if we have small size nucleic acids, these can also be separated on the acrylamide gel. Further, also I want to tell you that Agarose gel electrophoresis is preferably done vertically because elasticity of the acrylamide gel is much better than the agarose gel. Agarose gel cannot be used in vertical gel electrophoresis simply because of the poor elasticity, and because of this, if we want to remove the agarose gel from a vertical unit, it will lead to cracking and recovery of the gel becomes very difficult. So these are the few fundamental differences between the agarose gel electrophoresis and sodium dodecyl sulfate polyacrylamide gel electrophoresis. But as a routine matter, the acrylamide gel electrophoresis is used for the protein separation in electric field, and the agarose gel electrophoresis is used for the separation of the nucleic acids in electrophoresis. Thank you.